This video is on chromosomes, and in particular we're looking at the difference between the way the chromosomes are arranged in prokaryotic cells and eukaryotic cells. So really the main thing here is already outlined in the student outcomes. In eukaryotic cells we have a complex chromosome which is linear. The genome is divided into a number of linear segments called chromosomes, which are found in the nucleus and are bound to proteins. In prokaryotic cells we have that much simpler loop of DNA which isn't bound to proteins and isn't in a nucleus, and that loop of DNA sits in the cytoplasm. We also know from when we were looking at endosymbiosis that eukaryotic cells do have also some loops of DNA like this that are found in chloroplasts and mitochondria, and this is part of the evidence supporting the endosymbiosis theory. So prokaryotic DNA, one long loop joined up at the end. Eukaryotic DNA, a number of linear chromosomes in the nucleus bound to protein. And so this basically shows that in a different diagrammatic view. Now, note here that this view of what a chromosome looks like is probably different to what you're used to, and I'm going to talk about that a little bit further down. But basically, yeah, the chromosomes here, linear ones, if you're human, you're going to have 46 of these. They're just in a bit of a mess in the nucleus, and they need to be unraveled like this so that enzymes and other things can access that DNA. What we're used to seeing a little bit is this sort of wrapped up form of a chromosome, and I'm just going to make a really good point that this is not how they normally appear. This is only at certain times during cell division that you'll see chromosomes looking like this big sort of fat X kind of shape. So chromosomes are linear like this, and this is the condensed form. So this is the unwrapped one up here. When it condenses up, it ends up like this. Before cell division though, as we saw in the last video, the DNA has to be replicated, and this is the replicated form, and we can see that here, that these two sides of the chromosome, we call them sister chromatids. That's after replication. This is the one copy of the DNA, this is the other copy of DNA. So both of those, remember from the semi-conservative replication, you're going to get two copies of each chromosome when they get copied, and those two stay together, and then during cell division the chromosomes get sorted and separated out. So the reason the DNA is unraveled most of the time is it needs to be accessed and used for protein synthesis, which we talk about later. The reason it gets packed up during cell division is if you're going to sort out 23 pairs of chromosomes and have 23 go in this direction and 23 go in that direction, you don't want them to be a big unraveled mess. You want to roll those up and pack those tightly and make sure you know which ones are your copies so that you can put one copy in either daughter cell. So this view of the chromosome only happens for a short time just before they get divided up and put into two different daughter cells. So, as I said, for humans we have 23 pairs of chromosomes. So this is a microscopic image of those chromosomes. We have 2 copies of chromosome 1, 2 copies of chromosome 2, all the way through to chromosome 22. This is the microscopic view. This is a diagram showing how they look. They're named from the longest being number 1. And they get shorter and shorter and shorter down to 22, and then of course we have the sex chromosomes, which can be either XY for male and XX for female. You also notice here that there's some banding patterns, so depending on how this DNA is prepared and stained for the microscope, you can have those banding patterns. They also help people identify which chromosome is which when they're sorting them out and doing what's called a karyotype, where you look at the full set of chromosomes in a person. So why do we have 2 copies of chromosome 1 or chromosome 17, for example here, is that when you inherit the chromosomes from your parents, when there's a fusion of egg and sperm to create the first cell, the zygote, sperm has 1 copy of chromosome 1 and the egg has 1 copy of chromosome 1, and so those 2 copies are now in each of your cells as they grow and divide. And so we call these homologous chromosomes. Homologous means similar but not identical. And so the versions that you have from your mother and father of chromosome 17 will have the same genes in the same location, which is why they would have this same banding pattern, but they might not necessarily be exactly the same down to the base pair level. So there might be some variations or some mutations in one or other that make the DNA sequence different, even though they have the same genes in the same locations. So homologous chromosomes mean that you have 2 copies of every gene, one from your mother and one from your father. There are some videos down here, and if you want to look up notes on that, it's on page 6 to 8 of your textbook. Please let me know of any questions in class.